You are, yeah. You're
Okay.
set to go. All right, we'll let you rock and roll with it.
Sure. So I'm from the life sciences team, and so I'm going to talk today about life sciences and the intersection with storage and some of the implications. DDN and IBM had a very long strategic partnership, since 2007. Right now that we're working with the GRIDScaler product with the IBM Spectrum Scale file system, which was formerly known as GPFS. DDN provides some unique value-add features that are sanctioned by IBM, and it has some tremendous performance in terms of the file system. So I'm going to talk a little about, what is life sciences, genomics, what does that do with storage, and some of the problems and those sorts of things like that. So, what I thought at first I'd get started is with, let's say you're in some kind of a city, right, like Denver, and, you're going from one end to the other end, from start to finish. You want to visit some places or maybe any path as possible. And so this is sort of the classic city block traversal problem. You have a beginning and the end. You want a continuous, go north and east only. You don't want to repeat yourself in any kind of a cycle. So some questions that come up, right, are, how many possible paths are there in an n by n grid? And sort of paraphrase some David Byrne stuff, right? How did I get here? Right? How long is the path? Is there an optimal path if there's some certain things I have to visit? Are there weighted edges? How do we compute the path? What are all the different paths? So these are all sort of interesting questions, in this sort of classic city traversal problem. And so what does this have to do with life sciences? Well, it turns out actually it has some really interesting direct implication. So, when you have a sequence of pairs that you're trying to align, determining optimal base alignment for short sequence of base pairs. You can put these in a very similar type problem from one axis to the other axis, and then calculate using dynamic programming these batch and bound problems. And so there's a non-trivial way to find the best algorithm for scoring matrices. You can have weighted edges. And sort of these sort of questions arise of like, how did I get here? How do I find these kind of sequence matches? And this is a very important problem because if you take a look at all of these aligners and matches that do this, for anywhere from BLAST to Clustal to these other mechanisms, this is what they're using underneath, and this has some direct implications for I/O. Sometimes there's a little bit of a misnomer that sort of says, hey, all of my I/O that I do in life sciences is serial. Well, guess what? When you're actually matching these sequences, and I'm just showing you two very short sequences, but you can have things like you're comparing multiple genomes simultaneously across different species and different sections. And so you're doing this on different nodes that are accessing different parts of the different genomes, and so this becomes some very interesting I/O patterns, and it's important to have strong I/O performance when you have these sorts of things. Just as a note, V. Levenstein introduced this notion of the edit distance where you transform one sequence to another and so forth. But these are some of the sorts of algorithms that are underneath in life sciences that require lots of storage, but also require sophisticated I/O patterns and processing and those sorts of things like that. The Spectrum Scale file system does these sorts of things, and we've got some interesting things coming out where we're providing profiles to do genomics, to do life science optimizations that directly optimize parameters within Spectrum Scale if you're using it. And this is coming out with the newly released version of the Scale 5.0. I just want to give another simple example of another one where, in phylogenetics. And so let's say you have a sequence of numbers, right? 6, 1, 2, 3, 4, 5. And you're only allowed to swap a certain number of digits and to sort it. So, I go through this, on the top line, you see I do like a little swapping sequences. And I eventually get to the sort, and I do 1, 2, 3, 4, 5, 6. Well, again, this is important because, if you're looking at genomics, and this is another classic algorithm that's out there that uses genomics, and so the idea is that you're trying to say, hey, how does one sequence match up with another sequence? And so it turns out, for example, with comparing across species, this is called synteny. The mouse and human genome structures are actually similar, except there's about 300 subblocks that are rearranged like this. And as you can see, if you have long sequences, again, this leads to some very interesting, I/O patterns. And so, we want to get from one sequence to the other sequence with the most parsimonious or the fewest number of steps out there. And so, for example, like I show on the bottom, the same sequence above, if we could find another sort of way to sort this that results in a much faster sequence. And so this tells you sort of in the evolutionary tree how far species are from one another. Again, these are the sorts of algorithms that Broad and some of these other institutions are deploying, and these have direct impact on I/O. I should add something very interesting about this, is that one of the first important lower bound results that was done on this particular method of sorting by these sequences that really wasn't in the area of genomics at this point, but it was done by a professor, actually at that point he was a postdoc at Harvard, Christos Papadimitriou, and he had a very interesting co-author, and that co-author is Bill Gates, and that's the Bill Gates of Microsoft. He did this many years ago as one of his first results out there. So again, I'm going to just touch on this one, but another really important I/O impacting algorithm in life sciences is motif finding, and so motifs are, you're trying to find short, segmented portions that repeat themselves, but you don't know how long they are, where they are in the sequence, and this actually has some really critical type of factors. In there, these motifs are not known in advance, obviously, right? And so globally identifying and locating these things becomes important. For example, if you take a look at like some large mammals like whales and elephants and so forth, they have a much lower incidence of cancer. And you would think like a larger species would have more cancer, but the large physical body, they don't. And so it turns out, one of these things that they think is a regulatory mechanism is some motifs that repeat themselves that encode, how to find cancer. And so finding these types of motifs is extremely important in some different areas, and it can be very I/O and compute intensive for these sorts of things. You may have heard of this new technology called CRISPR. CRISPR, where you can edit genes and modify genes. And again, this is also the identification was based off and discovered off of motifs in there. It's a very interesting, both compute and I/O intensive type methodologies here that are used. So, what my point I'm trying to tell you is that, life sciences actually encompasses really a very broad spectrum of technologies and problems and solutions. And so we have sort of, on the left side from you on the screen, right, is sort of what I call the computational stuff. This is sort of the ab initio, this border between physics and physical chemistry type of things, where you're doing very intense compute type interactions, and you have the larger molecular dynamics, drug design, those sorts of things. Then you have the genomic stuff that I described a lot above. Sequencing and those sorts, and then you also have translational medicine, personalized health records, functional MRIs, and again, all of these have different compute and I/O characteristics. For example, like, in this translational medicine or functional MRIs, well, what's happening now in medical records and EMRs and so forth, when you go to your doctor, when they listen to your heart, they don't just listen to your heart and then that's it. A lot of there's physicians out there that now have recording stethoscopes, and they attach that to your medical record, and then they see how did that change over time, or you have some kind of a compute job that analyzes those things automatically across the medical records. And so these are all very interesting I/O management, deployment, scalability, performance, and all those sorts of problems out there. So really, the life sciences spectrum is really a very broad category of these sorts of problems, and that's what spectrum scale really tries to address. And getting back to sequencing, a lot of it has been driven by the fact that we've had a tremendous cost reduction in DNA sequencing. Back when the original Human Genome Project costed, it was like $1 billion, and now full genomes can be, sequenced and so forth, in the less than 10K. So there's really been a democratization of how sequencer our availability and accessibility, and so that's caused countries, states, city-states, local communities to say, hey, it's not just a human genome, but what are the plants, animals, microbial life, that is useful and important in my community that might have value, and I can figure out what these dynamics are, and so that's caused. Naturally, the cost per genome has dropped tremendously, but what that's also caused is an explosive growth of data. I'm showing, for example, some of the NCBI, the reference genomes, the accession database, and how that's grown. It's had this exponential growth. As the sequencing costs have dropped, many more things are being sequenced, and these sorts of things are being addressed. Again, this is a huge disk challenge. If you take a look at the, a 3.5-inch, 8 terabyte drive, I know there's 10 terabyte drives available now. We're looking at a 2,500 disk farm for like a 20 petabyte accession database that might be out there that people download for changes and comparison. And so, give you some perspective, right? You stack this horizontally. It's 127 meters high, and as a comparison, like the Eiffel Tower and the Statue of Liberty. So these are all very very large and complex problems that we're dealing with. Genomics at a very high level. I'm giving a rough pipeline, of we have the base calling from the sequencers to raw reads to the alignment BAM files to the variant files to finally the downstream analysis. And it really is a data tsunami that that's out there that's a forthcoming that many of you in your organizations have already seen and are probably dealing with as an issue. So where does Spectrum Scale really fit into this? Spectrum Scale is the file system that IBM's developed, and as I mentioned earlier, we've released version 5.0 today, and so the files, Spectrum Scale as a file system provides some really important criteria. Number one, it provides things like a single namespace. I sort of jokingly refer to it as that. We're not trying to solve these problems, at NIH or the pharmaceuticals. We as the tool vendors, DDN or IBM, are providing the plumbing, so to say, right? So we provide, for example, like you can access these file systems via POSIX type things, or NFS or SMB. You can connect to the sequencers. You compute the downstream results in the compute farm, and you have users. You can also, with Spectrum Scale, there's a Hadoop translation layer that will connect to it. And so, and behind the Spectrum Scale file system that it provides, are obviously things like the DDN storage machines. You can have tape. You can connect to cloud, and you can also do multiple sites. To interconnect between different sites, transfer information, and sharing of data. And so it's very important, right, obviously, to provide, ease of access, scalability. When you talk about these huge numbers of it could be some, for example, BGI in China is providing, has like 100 sequencers, and they're estimating that their growth per month sometimes is 30 petabytes per second from the sequencers that they're using running full time. And so, the ability to scale and deal with these sorts of large data are the sorts of things that Spectrum Scale excels at with these sorts of things like that, with a full comprehensive suite of protocols with which you can connect to. Some of the things that are important in Spectrum Scale that we've focused on, are simplification of management, so you can have a small number of, or one admin even deal with petabytes of data in a simple way. It has enterprise and flexibility features. It has erasure codes to protect and provide data integrity. You can have ILM, so you can go all the way from spinning disks to from SSDs to spinning disks to all the way down to tape. You can do multiple different sites and allow global collaboration type activity to occur. There's lots of different models of way you can deploy spectrum scale. You can deploy it, in a tight SAN network, in sort of a file server network, or even in a sort of a more classic, like a Hadoop style shared nothing type model out there as well. And as I mentioned before, there's a lot of ILM life cycle management that it provides. You can go to different types of storage pools and then come all the way back up into SSD or those sorts of things like that for the full information life cycle management. I know it's a busy chart, but again, I was just trying to show that, the life sciences, I sort of broke it out into sort of 4 different categories. Genomics, discovery, translational, data science. And nobody usually, unless it's a large organization or medical school, that implements a sort of full richness of this sort of spectrum of all of the different things that go from computational chemistry to translational medicine. But they usually, they choose some segment of this, and spectrum scale, and clusters and compute provide sort of the plumbing in between. And one of the things that I like to joke is, these arrows that you see connecting the relationship, right? I mean, they look small in size, but they're actually very important. I mean, they're the kind of stuff that discover blockbuster drugs or win Nobel Prizes or those sorts of things like that. Spectrum scale advanced file management is a way to do multiple different sites. Example of a different multi-site pharmaceutical. We have some major cancer centers that have different types of deployments of this. Again, this is used in personalized medicine. I was just going to give an example of where this young lady, she had a form of leukemia that they started to treat, but it turned out it wasn't treatable. And then they did a whole genome deciphering at Columbia University. And then in that process, they discovered that one of the cancer drugs, she had an immunity to it. Because of a certain defect called the Philadelphia chromosome defect. And they found actually that a kidney-related drug from Bristol Myers Squibb actually addressed this particular defect, and so they were able to, because of the genomic analysis, come back and add to her protocol regimen this particular cancer drug, and it sort of caused her cancer to go and rescind. So again, what I'm trying to say here is that spectrum scale and these sort of storage systems are really important because of the backbone that they provide, and they have a really collaboration, multi-site, single file system image or single namespace image across lots of different sites. So let me pause here and take some questions while I have some of the value slide up here for spectrum scale.
Any questions at all? Must have been so thorough.